Greetings everyone. This is Dr. Charanya, reader from Department of Conservative Dentistry and Endodontics, and today I am here to discuss on the topic biocompatibility of dental materials. So what does biocompatibility mean? Biocompatibility is that a material should be harmless to the pulp or the oral soft tissue which you're using. So it should be a material which is non-toxic substance, it should not induce allergic reaction, should have no carcinogenic potential and should exist in harmony with the tissues. So basically it should be compatible with the living oral tissues. Murray et al. In 2007 defined biocompatibility as the ability of a restorative material to induce an appropriate and advantageous host response during its intended clinical usage. Craig defined it as the ability of a material to elicit an appropriate biological response in a given application in the body. Going back in time, it was seen that Hippocrates, way back in 460 to 377 BC, did all ethical treatment and checked for safety and efficacy of any material before its clinical usage. But it was surprising to see that in mid-1800s, dentists started innovating newer materials, but they also started to directly use these on the patient's mouth. One such incident is the material which was developed by Fox, which is a combination of bismuth, lead and tin. It was directly poured into the cavity at 100 degrees Celsius. Even G.V. Black tested various new materials during the early period of amalgam preparations. This led to an unethical and illegal usage of materials directly on the humans. Thus, various organizations was developed such as FDA, ANSI, ADA and ISO. So the biocompatibility is basically an interaction between the body and the material. The interfaces between the material depends upon the location of the material, its duration in the body, its properties and the health of the host as well. The adverse effect of any dental material is evaluated based on the systemic toxicity, local toxicity, allergic reactions, which is the cross-reactive allergy or concurrent allergy, and other reactions which includes the genotoxicity, mutagenicity, carcinogenicity and tetrogenicity. Let's look into the first type of toxicity, which is the systemic toxicity. Systemic toxicity is that adverse reaction that appears on an area distant from the application site. That is, for example, if you give an overdose of local anesthesia, you might have an adverse effect in the CNS or the CVS. So this depends upon the duration and the concentration of exposure and also the site of exposure. Next is the local toxicity. Local toxicity is the response which is seen in the immediate vicinity of the contact with the substance. It is basically an unfavorable phenomenon where the material causes injury to the surrounding oral tissues, which is known as cytotoxicity. Cytotoxicity is nothing but the ability or the extent of destruction or killing capacity of a particular agent. The allergic reaction, which is of two types, that is the cross-reactive allergy or concurrent allergy. It is basically an antigen-antibody reaction. The cross-reactive allergy is that if you are allergic to a particular element, then it is assumed that he or she will be allergic to a chemically similar element. For example, if you are allergic to nickel, the similar chemical element is palladium. You might be even allergic to palladium. Second is the concurrent allergy. That is, two allergens which are frequently present at the same time can cause allergy to you. For example, TEC-DMA and HEMA, which is present in the composite, could be allergic at the same time. The other reaction includes the genotoxicity. It is that a material which can cause an alteration in the genome of the DNA. Mutagenicity is the ability of the substance to pass the genetic damages to even the next generation. That is, it alters the base pair of DNA, causing genetic damages even in the subsequent generations to come. Carcinogenicity is the ability of a material or a substance to release or induce a malignant or tumor. Tetragenicity is the ability of a material or a substance to cause malformation during the embryonic development. So how do we check the material is biocompatible or not? So Langeland in 1984 proposed a sequence which was later adopted by ISO. That is, the first includes the initial test, secondary test, followed by the usage test. Coming to the first, that is the in vitro test. Based on the various reactions, there are various in vitro tests which can be done. For systemic reaction, it is the direct cell culture. For local reaction, it is the direct cell culture, agar diffusion testing, filter diffusion testing, dentin barrier testing. The allergic reaction can be tested using cell culture models, and other reaction using Ames technique and micronuclease test. Coming to the animal test. Various types of animal tests are done to check the systemic reaction, which includes the LD50 and limit test. The local reaction is tested by implantation test. The allergic reactions are tested by maximization test and Buehler's test, and other reaction are tested using micronuclease test. The usage tests are performed in animals or in humans. The local reaction is basically tested using pulp or dentine test, the endodontic test, and also intraosseous implant test. So this is a common table which will give you a basic outview of various tests and its advantage and disadvantage. The in vitro test, though, has a major advantage of being quick to perform, least expensive, large scale screening, good experimental control and others. It has a major disadvantage of whether it is relevant when we use it in vivo conditions. The in vivo condition allows complex systemic interactions and is more relevant than the in vitro tests. But the major disadvantage of these in vivo tests is that it is more time consuming, and more importantly, it has large amount of legal and ethical concerns, and it is difficult to control and difficult to interpret and also quantify. The usage test has the advantage of having relevance of the use of the material, but the disadvantage again is that it is very expensive, time consuming, difficult to control, legal and ethical issues, and also difficult to interpret and quantify. So after we have completed the in vitro and the animal tests, it is sent to the FDA for approval. Once the FDA and ISO approves, then starts the clinical trials. That is, we use these material in the human. So let's see how the human tests are carried out. In phase 1, we get an informed consent from the healthy individuals, and the materials are tested and applied on the individuals to check its ideal or adverse reaction. After it is successful comes the phase 2, where this material is tested on a small group of patients with the aim to determine the therapeutic dosage of the material. Then comes the phase 3, where the same material is tested in a large scale, where sufficient number of patients are treated, and it is used to determine the acceptable dosage among the population. And phase 4 is that when we collect information regarding allergies and interactions with other materials and are reported to FDA at timely intervals to evaluate any delayed or adverse effects. Test programs for the biological testing of dental material is carried out in two progressions. One is linear progression and the other one is non-linear progression. Linear progression is that where the tests are carried out in primary phase, secondary phase, usage phase, and then it is acceptable for clinical usage. The non-linear progression test can be performed in two ways. That is, the primary, secondary and usage test can be combined and performed at the same time, or the primary test can be done followed by which a usage test, then a secondary test can be done. To conclude, we need to know that the biocompatibility depends upon the composition of the material, the location where it is placed, and its interaction with the oral cavity. It is mandatory for the clinician to know and understand the material that he is using and to get its best advantage. These are my references. Thank you.